Hi everyone, welcome to my YouTube channel Science in Home. In this video, I am going to show you how to estimate total amount of protein present in our everyday food samples by using a simple test known as BIURET test. For doing today's experiment, I will be using a solution of copper sulphate or blue vitriol. Also, I will be using a solution of caustic soda in water. For making aqueous solution of copper sulphate or toothpaste and sodium hydroxide or caustic soda individually, we will be mixing copper sulphate or blue vitriol with water and sodium hydroxide or caustic soda with water. Now we will simply be mixing these aqueous solutions. This is our final solutions of sodium hydroxide and copper sulphate. In previous video, I had shown you how to make paper from grass using sodium hydroxide or caustic soda. The link for my previous video is given in the description box. The main principle behind our experiment, the BIURET test, is that alkaline copper sulphate solution formed by mixing copper sulphate with sodium hydroxide will be reacting with any peptide group present in any food sample to give characteristic violet color. The more amount of protein present or peptide present, the more amount of violet color will be formed after the experiment. We will see the color change in the experiment. In water, copper sulphate or toothpaste will be breaking into Cu2 plus and SO4 2 minus, while NaOH or sodium hydroxide, also known as caustic soda, will be breaking into Na plus and OH minus. As we all know that proteins are made up of amino acids. Amino acids are composed of R group connected with C, which is connected with NH2 group and COOH. These amino acids are joined by peptide bond to form a peptide. These peptides are smaller chains of amino acids which are again joined by larger chain of peptides to form proteins. Our experiment will be checking the amount of any peptides present in any food samples by the reaction of Cu2 plus with the peptides present in the food sample, which will give a characteristic violet color. For today's experiment, we will be testing on three of the mostly used food samples in home. They are salt sample in solution, milk solution, and egg white, pure egg white. The yolk part of the egg is separated. Since it was getting evening and the amount of sunlight was decreasing, so we had to shift our experimental setup to artificial light inside our home. Take 2 drops of caustic soda. Then a small drop of copper sulphate. Now observe the change in color, which is not occurring. This is showing that salt solution does not contain any peptides or proteins. Seeing the amount of violet color given by these three solutions after BIURET test, we may conclude that salt solution does not have any protein content because it is colorless. On the other hand, milk and egg yolk both contains proteins or peptides. Thus, they have given violet color. After shaking the milk and egg yolk solution, we can conclude that the amount of protein present in egg yolk is far more than the amount of protein present in milk. Thus, milk have given fainter violet color than egg yolk. Thank you for watching my video. If you like this video, then kindly subscribe my channel for getting more content like this in future. Thank you.